Hello, everybody. Uh, this is Suman Chakravarti from Multicase, and it is my great pleasure to introduce Dr. Naomi Krulok, the lead scientist from Center of Drug Evaluation and Research, US FDA. And she has worked for US FDA Center for Drug Evaluation and Research as a computational toxicologist for 18 years, developing and applying QSAR models to support the regulatory review of pharmaceuticals. She is the scientific lead for CDER's computational toxicology consulting service and is the principal investigator of 3 FDA CDER RCA collaborations with commercial software vendors. Dr. Kurlok has published extensively in multiple journals for peer-reviewed articles describing data standardization, transformation, and classification for modeling purposes. And she holds a BSc and a PhD degrees in chemistry from the University of Salford, England, and the University of Calgary, Canada, respectively. On a personal note, I have known Dr. Krulok for more than 15 years and had the pleasure to work with her during this time. I have known her as a first-class scientist and very knowledgeable, yet very approachable. I believe she and her fantastic team established QSAR as an important tool for regulatory decision-making in FDA, and as a result, uplifted the QSAR field, which benefited us all. I would like to thank her for graciously agreeing for this presentation and looking forward to learn from and listen to it. I'm also thankful to my colleague Gianna Cioffi from Multicase to work tirelessly to arrange this webinar, and Mr. Etindra and Monarajan Sidhu ji from Neotel to make this possible. So without further ado, here is the presentation from Dr. Naomi Krulok.
Good morning. I'd like to welcome you to my webinar today on the application of QSAR and expert knowledge for ICH-M7 impurity classification. My name is Naomi Krulak, and I'm the scientific lead for the Computational Toxicology Consultation Service at the US Food and Drug Administration's Center for Drug Evaluation and Research. This is an outline of my presentation today. I'm going to start with an introduction as to why we use QSAR, or Quantitative Structure Activity Relationship Modeling, to assess the safety of drug impurities. I'll then move into some of the basics of the QSAR methodology, starting with how we go about building a model, and then some considerations with regard to applicability domain, as well as structural limitations of the methodology. And then I'll finish up this section by looking at the use of the generalized published structural alerts to QSAR models for predicting the endpoint of mutagenicity. I'll then move into the focus of this presentation, which is the ICH-M7 guideline, looking at the specific recommendations on how QSAR should be applied. I'll cover the OECD validation principles to demonstrate that a model is fit for purpose, and then the application of expert knowledge to improve the robustness of QSAR predictions. I'll then discuss different ways to search for structural analogs that can be used to support or refute a prediction, approaches including substructure searching and global similarity searching. And then I'll move into some case studies that show how we can apply expert review to resolve conflicting predictions, as well as to resolve out-of-domain results. I'll then finish up with some considerations with regard to how we present the information in a regulatory submission and report that information comprehensively, and then some concluding remarks. So let's begin by looking at the safety assessment of drug impurities. Well, drug impurities, unlike an API, offer no direct clinical benefit to a patient. They're going to be present regardless of the control strategies that are applied. By their nature, some of these impurities are going to be reactive molecules, and therefore they have the potential to be mutagenic. And of course, mutagenicity is tied to the multi-step process of carcinogenicity. The effects may not be evident in patients for many years, and it's not going to be an effect that's identified through clinical monitoring. So evaluating the mutagenic potential of drug impurities is an important component of safety assessment. But we recognize that from a practical standpoint, although a cautious approach is warranted, conducting an empirical assay for every impurity may not be feasible or even justified. So we need to come up with some impurity evaluation process that balances that need for high throughput with the regulatory imperative of maximizing patient safety. So that's where QSAR comes into play. QSAR provides the high throughput process needed to handle a large volume of impurities, and is demonstrated to have adequate sensitivity for predicting bacterial mutagenicity, which is critical for patient safety. For impurities, QSAR is now considered fit for purpose for impurity assessments under ICH-M 7. It's recommended by regulatory agencies, and it truly is a state-of-the-art approach to assessing mutagenicity. So now let's look at some of the basics of the QSAR methodology. What is QSAR? Well, QSAR stands for Quantitative Structure-Activity Relationship, and it's a type of modeling that identifies associations between attributes of chemical structure and biological activity. And in the context of this presentation, that activity is mutagenicity. It works under the general assumption that similar molecules exhibit similar chemical and biological properties, and therefore that toxicity can be explained by chemical structure. The modeling process itself learns from the results of actual laboratory testing. And so we use a computer to look for pieces of chemical structures that are statistically associated with a positive response in the Ames assay. These structural pieces are then set aside and labeled as structural alerts. But the modeling process can go a step further to identify other structural attributes that have the tendency to mitigate activity. So if we have a structural alert that's present in a molecule, but the molecule is empirically negative, what is it about that molecule? What are the other features that are present that are causing that structural alert to be negated? And the computer modeling algorithm has the ability to look for those trends and identify mitigators as well as structural alerts. Once we've identified all of these alerts and mitigators, we can save that information as our model and then use it to make a prediction of a new chemical's toxicity based solely on its structure. And this, of course, is very helpful in cases where we have a data gap. We have empirical data that is lacking for our impurity, and we need to make a prediction. And just to clarify this point for future slides, the term QSAR refers to a quantitative relationship that's typically seen in statistical-based models, whereas SAR refers to a qualitative relationship, which is most often seen in expert rule-based models. But collectively, we refer to them as QSAR with the Q in parentheses. So how do we go about building a QSAR model? There's really three things that we need in order to build our model. The first is a set of chemical structures that are represented in a format that is readable and interpretable by a computer, and that's through the use of molecular descriptors. And those descriptors essentially create a fingerprint for each of the chemical structures that captures information about the substructures that are present. For each of the chemical structures in our training set, we also need to have some kind of numerical score that indicates the activity of that chemical in the endpoint or the assay that we're trying to model. So in this case, we're talking about bacterial mutation, which is tested with the Ames assay. So chemicals that have been tested and shown to be negative will get a 0, and chemicals that have been shown to be positive will get a 1. And then the last thing we need is an algorithm to extract the patterns and trends between the substructures in the chemical structures and the activity scores. And this is our computer algorithm that basically breaks down all of that information to look for those structural alerts and mitigating features. And then it can store that information as a QSAR model. So now we come along with our impurity structure that we would like to generate a prediction for. The first thing the model identifies is that this molecule contains a structural alert in the form of a primary aromatic amine, but it also recognizes that there are substituents on either side of that amine in the ortho position that are going to be mitigating features. So collectively, the model will generate a prediction, in this case is negative, because it believes that the mitigating features are going to overrule the effect of the structural alert. For the QSAR methodologies, there really are two generalized categories that most models fall into. The first is statistical-based models, and the second is expert rule-based models. Statistical-based models are those that are generated through machine learning using a classic training set. And some of the examples of the algorithms that are commonly used are shown here, including partial least squares regression analysis, support vector machines, discriminant analysis, or Bayesian. These types of models are generally very rapid to build, but they can vary quite widely in their interpretability, both with respect to the way that the model is making the prediction, but also because of the way that the model is interpreting chemical structure. And so that's an important consideration when one is looking for the type of model that is most suited to an impurity assessment under ICHM 7, where we really want to be able to understand the underlying reason for a prediction, not just get a yes/no answer. In contrast, expert rule-based models are very interpretable. They are intended to capture human expert-derived correlations, and they're often supported by very rich mechanistic information and citations. When we get a prediction from an expert rule-based model, we understand what the alerting moiety is, but we can also be given by the model itself information on the reactivity of that moiety, how it would actually react with DNA to cause a mutation, and then supporting citations as well as structural analogs for that prediction. Expert rule-based models have the benefit that they can also anonymously capture knowledge from proprietary data without those proprietary data needing to be included in the model itself. And so that brings to the user a lot more information in a usable fashion. The downside to expert rule-based models, however, is that they're very time-consuming to build, and some of the best examples that are available have taken many, many years to reach a point where they are adequately predictive and comprehensive. The QSAR software used by us at FDA CEDAR comes from 3 different companies, and that software is made available to us under formal research collaboration agreements. Each company has both a statistical-based model as well as an expert rule-based model. And the specific models that are highlighted in red are what we use for our standard analyses. We have access to all 6 models, but in reality, we don't really need predictions from all of them for typical impurity assessments. We may just look at those other predictions in cases where we're having difficulty resolving conflicting calls, or it's a particularly unusual type of impurity that we're evaluating. We've chosen these QSAR software to work with because they use different methodologies, both with respect to the algorithms that are used to build the models, but also the way that the chemical structures are interpreted. And this means that the predictions are complementary. They sometimes agree with one another, but sometimes they don't. And that's because each of the models have their own respective strengths and weaknesses. So we can benefit from this by using the models in combination to increase sensitivity and negative predictivity by using a rule where if any one of the models calls a prediction positive, then we'll go with a positive overall conclusion. And that's consistent with the way that the ICHM 7 guideline is written. Additionally, we run a second statistical system to improve overall coverage. And what that means is that in some cases, one of the models may be unable to make a prediction because it doesn't have enough information about that particular impurity, but another of the statistical models is able to make a prediction. So by running a set of 3 models as our first initial standard assessment, we increase the chance that we will at least get one expert rule-based and one statistical-based prediction that's valid, that is consistent with the ICHM 7 guideline. Another reason that we use these particular software are because their predictions are chemically meaningful and transparent. So when we get a prediction, we don't just get a yes/no answer, we get an explanation as to why that answer was made. So the structural alerts that are being identified to support a positive prediction are reported by the model in its output. And in addition, we as expert reviewers have the ability to drill down and look at the underlying training set structures that alert was derived from to confirm that they are actually relevant to our impurity of interest. This really facilitates the application of expert knowledge, which, as I mentioned, is an important piece of the ICH-M7 guideline QSAR recommendations. And then lastly, we work with these particular software because they're publicly available. The models are trained on data that are from the public domain, so we can see the specific training set structures behind a prediction. And it also means that our results are reproducible by pharmaceutical applicants. We're all running the same versions of the software, and we'll all get the same answer. And so that puts everyone on an equal playing field. Applicability domain is an important consideration when we are looking at QSAR predictions. The applicability domain of a model is the region of chemical space within which that model can make predictions with a given reliability. And for each of the software that we work with, there is a definition of what is considered an applicability domain within which we can accept predictions, and outside of that domain we would consider those predictions to not be reliable enough to be able to use for regulatory purposes. The chemical space is defined in each different software using the structural attributes and properties of the training set molecules, but the different software do use different methodologies for considering those structural attributes and properties. For example, some models may consider physical chemical descriptors, and if we look to this figure that I have on the left-hand side, we can see that the test chemical has physical chemical parameters that are outside of the range of the molecules that were used to train the model. So this is a case where that test chemical may not be adequately covered. Other methods use molecular fragments. So in this case, the model will check all of the fragments that are present in the impurity and make sure they're actually present in some of the molecules that the model was constructed from. And if any of those fragments are deemed to be unknown, then the model will flag that and will indicate that the molecule, the impurity, is outside of the applicability domain and no prediction can be made. And then lastly, another method that's often used for assessing applicability domain of models is global similarity. And this is where the model will check the overall similarity of the impurity being assessed to the training set structures to confirm that there are at least some structures that are within a similarity threshold. So they're sufficiently similar to support a prediction. So different models have different ways of interpreting coverage. And in fact, even models that use the same general method, for example, fragment-based statistical models, can differ quite widely in their coverage. That's typically because the models are using different ways to interpret chemical structure. So their fragmentation may be different, but also their training set structures are likely to be different. So there's just different knowledge that's being used to build the model. So what this means is we can use that to our advantage to obtain a valid prediction. If we have to run one expert rule-based and one statistical-based model, but then when we run that statistical-based model, the model considers our impurity out of domain, we would then need to find some way to fill that gap. And so our internal practice is to use another model, and we actually run that set of 3 models together to ensure that at least one of those statistical models is likely to generate a valid prediction. So it minimizes rerun time. I think an important message here, though, is that if you run multiple models and they all give out of domains, then that really means that you may not have enough information to be able to give a QSAR prediction. It may be that some other type of approach to handling that impurity is going to be more appropriate than QSAR. There are some structural limitations of the methodology. This type of methodology, the QSAR methodology, is really intended to predict organic molecules that are on the smaller end of the molecular weight scale. They're generally not intended to predict molecules that are over about 1,000 Daltons, where three-dimensional parameters such as folding and other physical chemical parameters may come into effect. Those effects are not going to be captured by the model, so the model is not going to be able to adequately consider those when making a prediction. Inorganic molecules such as simple inorganic salts or coordination compounds are not going to be adequately predicted. And in the case of anything that has metal centers, particularly transition metals where you can have multiple oxidation states with different chemical properties, we're just not going to have the body of information in our training set to really adequately predict those types of classes. And then, of course, organic compounds that are mixtures. We're not able to make a prediction of the mutagenic potential of a mixture. We can predict the individual components, but we don't necessarily know what the outcome will be if the mixture is tested. And of course, anything that's not covered, anything that has a unique feature that is not represented in our training set is not going to be adequately predicted by the model. And then the last point I'd like to make is that the methodology really does not differentiate between stereochemical or geometric isomeric pairs. And that's important because what it means is if you run a prediction on a pair of isomers, you're going to get the same answer. And furthermore, if you run a prediction on an isomer where the opposite isomer is actually in the training set, the model will think it's an exact match or a duplicate and will just report back the information for the opposite isomer, which is not quite the same as a prediction. So a question that often comes up is, why do we even need to use a computer to do this type of analysis? There are many published structural alerts in the scientific literature, and why can we not just use those with visual inspection to identify molecules that are likely to be mutagenic? And the answer to that is that highly complex associations can be captured by a model that are not necessarily going to be represented in a published structural alert. The alerts that we find in publications are generally quite broad, and they do not consider mitigating features or the cumulative effect of multiple substituents. So an example of that would be primary aromatic amines, which are shown with this example on the right-hand side. The mechanism for reactivity is through formation of the nitrenium ion at the nitrogen, which then forms an adduct with DNA. And the ability of the molecule to form an adduct is highly impacted by both electronic and steric factors. So if we can stabilize the nitrenium ion by adding electron donating groups around the ring, we'll increase its mutagenic potential. However, if we introduce steric bulk on either side of the amine in the ortho position, we're likely to reduce its ability to actually get in close proximity with DNA and create an adduct. And these types of patterns were captured very nicely in a publication by Alberg et al. In 2016. I'm showing a portion of one of the tables in the bottom right-hand corner, and it shows the differences that we see across the different substituents and where they appear on the ring. So in some cases, we can have a substituent that is strongly activating when it's in the para position, but it's strongly deactivating when it's in other positions. So to illustrate this point, I'd like to share with you some data that we generated over a 2-year period from 2014 to 2017, where we compared the predictions that we get from QSAR with predictions we would make based on published structural alerts. So in this case, we had 900 structures, or just over 900 structures, that all contained a published structural alert. And what we found was after we applied QSAR and expert knowledge, a whopping 35% of those structures we would consider to be negative for bacterial mutagenicity, just because the models, combined with expert knowledge, are taking into consideration many more of those factors that you wouldn't get from just the presence of a structural alert in the first place. So some of the common false positive motifs that we identified, which are actually consistent with those that were reported by Maiden et al., are epoxides, carbamates, alpha, beta, and saturated ketones, where in each case the effect, the type, position, and size of the substituents greatly affects the mutagenic potential of the molecule. So I think this underscores the value of QSAR as a more refined approach to predicting activity based on all aspects of chemical structure. So now I'd like to move into the ICH-M7 guideline, which is the focus of our presentation today. The guideline was published in June of 2014 and revised to version R1 in March of 2017 to include an addendum that contains compound-specific acceptable intakes for common impurities. The title is Assessment and Control of DNA Reactive or Mutagenic Impurities in Pharmaceuticals to Limit Potential Carcinogenic Risk, and it describes how a hazard assessment should be conducted on a pharmaceutical impurity and how it can be assigned to one of 5 impurity classes. Table 1 describes the 5 classes that an impurity can be assigned to. Classes 1 through 3 are where we would assign an impurity that's going to be treated as mutagenic, and classes 4 and 5 are where we will assign a non-mutagenic impurity. Classes 1 and 2 are reserved for assignments based on experimental data. So in the case of class 1, these are compounds that are known mutagenic carcinogens, whereas class 2 is for compounds that are known mutagens based on a positive response in the Ames assay. Class 3 and 4 are for assignments based on QSAR. So a class 3 will be for impurities that are predicted to be positive based on the presence of a structural alert as identified through a QSAR prediction. Class 4 is where we can assign an impurity that contains a structural alert as identified by QSAR, but that same alert is shared with an empirically negative compound where the alert is in the same chemical environment, and therefore the alert can be dismissed and the impurity can be treated as non-mutagenic. And then lastly, class 5 is where we can assign any impurities based either on negative experimental Ames data or a negative QSAR prediction where there's a lack of structural alerts. And I should note a couple of points. The terminology structural alerts in Table 1 is referring to alerts identified by QSAR, not alerts identified by visual inspection. And therefore, a class 4 or 5, so a non-mutagenic assignment, may not be based on visual inspection alone. Section 6 of the guideline outlines exactly how QSAR should be applied under ICH-M7, and it states that 2 methodologies that are predictive of the outcome of the bacterial mutagenicity assay should be used. Those 2 prediction methodologies should be complementary to one another. One should be expert rule-based, and the second should be statistical-based. And furthermore, the prediction methodologies that are being used should follow the general validation principles that are set forth by the OECD. It goes on to say that the absence of structural alerts from those 2 complementary methodologies is sufficient to then conclude that the impurity is of no mutagenic concern and no further testing is recommended at that time. The OECD validation principles outline 5 pieces of information that should be associated with a model that's going to be used for regulatory purposes to show that it is fit for purpose. One of those pieces of information should be clarity around the endpoint that the model is predictive of, and that is based on the model training set. So for example, if we want to have a model that's predictive of bacterial mutagenicity in the Ames test, that model needs to be trained on all 5 strains of bacterial mutagenicity rather than just, for example, TA100. So that information needs to be documented in the model somehow. Secondly, there should be clarity around the algorithm that was used to generate the model, and that allows regulators to understand whether the model is a statistical-based or an expert rule-based model. There should be a defined domain of applicability. It's important for the user to know whether the model is adequately covering a given impurity, and it needs to alert the user when an impurity is outside of the applicability domain and that prediction is not going to be reliable. There should also be appropriate goodness of fit, robustness, and predictivity measures, which can be assessed through standard validation procedures like recall or cross-validation or external validation. And lastly, it's ideal to have some sort of mechanistic interpretation of the model. An important component of the ICHM-7 guideline is the provision for the application of expert knowledge, which will allow us to provide additional supportive evidence on the relevance of any positive, negative, conflicting, or inconclusive predictions, and that provides additional evidence to support our final conclusion. So as an example, this is how we would go about applying expert knowledge to our impurity. The model prediction in this case comes out as being negative. But if we drill down into the model output, it should tell us that there is a structural alert that's been identified, and it will tell us what the underlying training set structures are for that alert. It should also tell us if there are mitigating features that are present that in this particular case are actually overturning the presence of that structural alert and leading to a negative prediction. It's important for us to be able to take a look at, again, the underlying training set structures from which the mitigating features were derived to make sure that they are sufficiently similar and relevant to our impurity that it's likely that our impurity has its alert mitigated. So all of these things require some knowledge of mechanism of reactivity, and that's what the expert is able to bring to bear on this evaluation. So in this case, we can think about whether these alerts and mitigating features make sense. We know that primary aromatic amines are a class of mutagens. We know that they react through formation of the nitrenium ion at the nitrogen. And we also know that steric bulk in the ortho position on either side of that nitrenium ion is likely to reduce the ability for DNA adduct formation. So this prediction makes sense to us. So we would likely agree with what the model is providing us as an explanation in this case. We do have the ability to drill down and look at training set structures that are supporting alerts as well as mitigating features. And this is often provided in the model output itself. Most of the widely used software tools actually provide an analog table where you can go through and actually look at the training set structures to use that information to support your prediction or refute it, perhaps in some cases. But you also have the ability to bring in information from other structurally similar compounds that may not have been used to build the model. You may have in-house databases of your own with toxicology results that you can also bring in to support or refute your QSAR model conclusion. And the identification of the most important or relevant analogs is done through supplemental searching of databases using structure-based search techniques. And that's what I'll talk about next. So let's now take a closer look at structural analog searching techniques. So as I mentioned, searching for structural analogs and using the information from the analogs allows us to come up with a more robust conclusion. And this is what the application of expert knowledge allows us to do. Analog searching will allow you to confirm a prediction, but it may also lead to you refuting a QSAR prediction and overturning it for a different conclusion. Structural analogs are generally used in combination with mechanistic knowledge. When you're reviewing the analogs that you get from your search, you need to understand which are relevant because they have the same type of mechanism of mutagenicity as you have in your impurity. This approach can add confidence to predictions. It can also provide evidence for dismissing an alert, and it's particularly useful in cases where you need to resolve an equivocal prediction. If you have a weak positive prediction or you have conflicting calls from multiple models, structural analog review can actually be very helpful in pushing that prediction to a clear positive or clear negative. It's also very helpful in cases where you have an out of domain, you have a data gap. You can use structural analog searching of external databases that were not used to train the model to sometimes fill those gaps and come out with a valid prediction. Bacterial mutagenicity is driven by DNA reactivity, so the most relevant analogs are going to be those that have local similarity to a structural alert or a suspected DNA reactive feature in your impurity, rather than those that are globally similar, so based on the whole molecule. Most QSAR models do provide analog tables for you to review as part of your expert review process, and that's in their prediction output files. But you also have the ability to independently search a toxicology database using chemical structure, and that can be done using substructure search queries or global similarity search queries, and I'll walk through a couple of those in a moment. Once you have your set of analogs that you've identified as being structurally relevant, you can then drill down and review the actual data that's associated with those analogs and make sure that the data themselves meet your acceptance criteria. So let's first walk through a global similarity search for a query molecule. This is our same impurity that we've used for some of the previous examples. And when we get our series of hits back from querying this molecule, we'll actually get a similarity index that's calculated for each of the hits. And that will tell us the degree of similarity between the hit and the query molecule. That is most often calculated using a Tanimoto index, which is a simple equation that basically expresses the degree of shared fragments between the query molecule and the hit. So the higher the index value, the more similar the molecule is. And these values are usually expressed as ranging from 0 to 1. So in these cases, we can see that we have 3 negative examples, Ames negative results that have reasonably high similarity, and then a couple of lower ones, one of which is positive and one of which is negative. So our job is to decide which of these are the most relevant to our query molecule. If we look at the alert environment in the most similar examples that we were able to pull back, we can see that it's actually very similar to the alert environment in our query molecule. We have the primary aromatic amine, and it's flanked on either side by substituents. And in fact, we can certainly tell just by visual inspection that this first example is almost identical to the query molecule, and we know it's empirically negative. So that's a very informative data point for us. And then as we go farther down the list, we can see, well, if we even have substituents on one side of the amine, it's still negative. So that's also very helpful because it's saying really you just need to have that steric hindrance on one side, and we still have a lack of a mutagenic response in this particular case. When we look down a little bit deeper, we can see that there's another example which does still have some degree of similarity, although it is notably lower. And this example is actually positive. But what we need to think about is whether this mechanism of mutagenicity is relevant to our query molecule. And in this case, we find that this molecule is actually its own class of mutagenic compound, an anthraquinone, and that's going to react by a different mechanism than nitrenium formation. So we can dismiss this as not being a relevant structural analog. And then lastly, something we have to be careful of when we're doing similarity searching is we can get hits that actually don't even contain the structural alert of interest. And at that point, we need to dismiss those. This compound at the bottom at 0.47 is empirically negative, but it doesn't have the alert. So while it might tell us something about the backbone of the molecule being negative, it doesn't have any information about the nitrenium ion formation and DNA adduct formation. So in that particular case, we would also dismiss it. Now, substructure searching for analogs is a slightly different approach, where we're searching for a piece of a molecule rather than the whole molecule. So I'll go back to my original example of primary aromatic amines. This is the class that our impurity falls into, and so we're interested in comparing our impurity to other examples that contain that same moiety. Now, something to be aware of is, in the case of a primary aromatic amine, it's a very common fragment that we find in publicly available databases. So if we do a search by just drawing aniline, the search query is actually going to give us almost 900 hits. Many of these are not even closely relevant. So they have an amide, or they have other functional groups where the amine is not even in a primary environment. So we may need to refine our query. So we can do that in this case by explicitly adding the hydrogens when we draw the molecule before we do the search. And that reduces it now to 282 hits, where at least now we're only getting examples where the amine is terminal. But we can see, but just looking at this small subset of hits that we get back, these are still not the most relevant examples. And furthermore, we still have 282 to skim through to find the best examples. So we want to refine it further. So the next refinement we can do is to add substituents in the ortho position, because we know that that's important for this class of mutagenic compounds. And in this case now we only get 7 hits, so we're getting a much smaller set of compounds back for us to actually review. When we look through the compounds and review the activity of those compounds, so the Ames mutagenicity status of those compounds, we see that 3 of them are negative and there's 1 example that's positive. However, if we look at the steric bulk of those substituents in the positive example compared to our query structure, we can justify that our impurity is likely to have much more steric hindrance. So this positive example is probably not likely to be enough to sway our decision and call our impurity positive. In contrast, when we look to other more bulky substituents, and of course this is the same hit that we got through the global similarity searching a moment ago, we now get a negative response. And so this is again much more useful information. So I think the important thing to take away from this demonstration is that we can really refine the query to identify the most relevant analogs. And it's often helpful to start in a more generalized fashion and then add pieces or make the query more and more specific until we get down to a small set that are really relevant. And then as a reminder, bacterial mutagenicity really is driven by DNA reactivity. So local similarity to the structural alert is going to be more important than global similarity. And of course, the most globally similar analog may not always be the most relevant. In the example I showed a moment ago, it is a relevant analog. It's highly similar and it's almost identical. But it's certainly possible to have a globally similar analog with a very high index that still lacks that structural alert that's important. And in that case, you would need to dismiss that structural analog. We sometimes encounter situations where it can be difficult to find a structurally similar analog to our impurity. In these sorts of situations, one way that we can fill this gap is to make substitutions. So rather than searching for a piece of the molecule, we can search for something that's highly similar to it or that has similar electronic or steric factors. And this can be particularly useful in cases where we have an out-of-domain result due to an unknown fragment. A piece of the molecule may be unknown to the model and may not have any representation in the model training set. And there's a good chance that even if we search external databases, we may not find molecules containing that fragment. And so we can modify the fragment and then search for those modified analogs. And those are often adequate to support a valid prediction. So the types of substitutions that are typical are to find examples that have similar leaving groups. So in the case of a halogen, if we have an impurity that's a bromo analog, we might search for the chloro analog or the iodo analog. We can also, in cases where we have an electron donating group, substitute that group for other similar groups. So in the case of an alkoxy, we might substitute it for a secondary amine. Similarly, for electron withdrawing groups, we might substitute a sulfoxide for a carbonyl. When it comes to aromatic rings, we have a lot more options available to us. So if we have a pyrimidine in our impurity and we're having a hard time finding analogs that are similar to that, we might redraw that analog with a pyridine and search for pieces of the impurity with that modification. Or in the case of 5-membered rings, we might have an oxazole and we might replace it with a thiazole, for example, or a furan. So now I'd like to walk through some QSAR evaluation case studies to demonstrate how we handle different prediction scenarios. The first case study that I'd like to present is how we can reconcile different predictions across the multiple models that we're using. And in this particular example, we actually have experimental data, although it's limited. And that kind of adds an extra layer of complexity to the mix. So we see that our statistical models are flagging the aromatic amine as alerting, but one of the statistical models gives the prediction as positive based on this amine, whereas the other considers partial mitigation and downgrades that prediction to an equivocal. We know that there is limited strain negative data available already, but we note that this is in TA98 and TA100, and so by itself that's not adequate to dismiss the positive prediction that we're getting from the statistical based model or the equivocal prediction we're getting from the other model. So we reviewed a number of structural analogs that contain the primary aromatic amine in a very similar environment with similar electron donating and withdrawing properties in the same positions around the ring. And we found that we really didn't have sufficient evidence to overturn the positive prediction. And so what this means is, as a regulator, we will take the more cautious position and say, because we cannot justify with good solid scientific evidence overturning that positive prediction, we're going to call this impurity positive and assign it to class 3. And as an anecdotal update, we actually found that when the impurity was tested in a full 5-strain Ames, it did actually give a positive result. So in this particular case, our cautious approach was appropriate, and we adequately and accurately characterized this impurity as mutagenic. The second case study I'd like to present illustrates how we can handle an out-of-domain result. And this is a prediction scenario that comes up quite frequently for new drugs, where one of the models that we're using to make a prediction actually generates an out-of-domain result rather than a valid prediction. Now, in the case of the parent drug itself, we have negative empirical Ames data available, but the problem arises where we have a late-stage impurity, which is highly similar to that parent drug. It also gets an out-of-domain result from one of the models because it shares those same unknown features. And in this case, we don't have empirical Ames data to support an assignment. So this is where we don't have enough information to assign that impurity to class 5, and so we need to use expert knowledge to fill that data gap. So we can consider the impact of that additional moiety in the late stage impurity and whether it's going to increase the mutagenic potential of the molecule. In this case, we can conclude that it's a hydroxyl group, it's highlighted in red, and it's not likely to be DNA reactive, and therefore it's not likely to increase the mutagenic potential of that molecule over the level of the parent drug itself. So we can use the Ames data from the parent drug to support an overall negative or class 5 assignment for the impurity. And then the last case study I'd like to present is that of an assay artifact. And this is a situation where we have an acid chloride and we're getting an equivocal prediction from our expert rule-based system as well as one of our statistical-based systems. So this is kind of a weak signal. Further investigation into this class of compounds shows that they often yield false positive results when tested in DMSO due to the fact that the alkyl or the acid chloride actually reacts with the DMSO to form a mutagenic substance. And so this information is based on a paper by Amberg et al. And when they compared testing of a large number of acid halides in both DMSO as well as other solvents such as acetonitrile, almost all cases ended up with a negative result in the Ames assay when they were tested in a non-reactive solvent. And so in this situation, we can apply that knowledge to support an overall negative prediction or class 5 assignment. And of course, from a practical standpoint, acid halides are effectively purged with water. So it's actually unlikely that this type of an impurity is going to be finished in the drug product. So the last section of my presentation covers comprehensive reporting of QSAR results. There are several components that are important to include in a well-documented QSAR analysis. The first is a materials and methods section that lists the software that has been used for the analysis, as well as the specific models within that software and their version numbers. Secondly, there should be a table that includes the individual model predictions as well as the overall classification that's being assigned for each impurity. Then there should be some detailed explanation of expert knowledge if it's being applied. And this is particularly important in cases where expert knowledge is being used to overturn a prediction from the QSAR model. We need to be able to understand the rationale for changing that conclusion. And then lastly, it's helpful to have an appendix that contains the raw model output files. And that gives us as regulators the ability to go back and make sure that the conclusions from the model have been adequately interpreted and transposed into the table. Additionally, this appendix can include any pivotal experimental data that may be used to support an overall classification. So this is an example of how that information might be presented in a table where we have the chemical structure as well as the chemical name, and then we show the individual model predictions, in this case an expert rule-based and a statistical-based, as well as the overall ICH M7 classification. So just walking through these examples and how they relate one to another, we see that in the first example we have an aromatic nitro alert. But we also see from the comments field that that alert is found in the same chemical environment as the last example, chemical 4. And chemical 4 has been tested in the Ames assay, and the appendix contains the data for that result. And so due to the fact that we know that chemical 4 is class 5 due to the negative result in the Ames assay, we can dismiss the structural alert that is being identified by both the expert rule-based statistical models. And we can apply that information to chemical 1. So this now chemical 1 is assigned to class 4. In the case of chemical 2, we have negative predictions from both the expert rule-based and statistical-based systems. And this is not unexpected given that we've already dismissed the nitro group in chemical 1, and we know that corresponding amines tend to be less mutagenic. So if we know the nitro group is negative, and it makes sense that it may be due to the fact there's ortho substitution, it's very likely that the corresponding amine is also going to be negative. So based on our expert review, this prediction makes a lot of sense. And then for chemical 3, we see that we have the nitro group once again, but this is in a slightly different chemical environment because there's no orthosubstituent on one of the sides, which means that that steric bulk is reduced, and therefore it's not unreasonable to assign this to class 3. And even though we get an out-of-domain result from the statistical model, at this point it doesn't matter because the fact that we get a positive from one of the systems is going to be sufficient to assign this impurity to class 3. There are a few special considerations with regards to reporting and interpreting QSAR results, and one of those considerations relates to software versions. We prefer to see predictions made with the most recent version of any QSAR model. That represents the current thinking and the most up-to-date knowledge. But we do recognize sometimes it's not practical to update predictions in a submission, perhaps because the version update occurred very recently. And so old predictions may be acceptable unless the conclusions are questionable. And a situation might be that there's a visual structural alert in the molecule, but the prediction came back as being negative. And so that's something we may look at just a little bit more closely to confirm that we agree with that negative conclusion. We consider QSAR predictions internally to have a 2-year shelf life, and this is an unofficial rule of thumb. And so what that means is when we start to see predictions outside of that 2-year window, we're more likely to look at them more closely. Another consideration is with respect to out-of-domain results. An out-of-domain result is not a prediction, and it does not contribute to a negative or class 5 classification. And what that means is if you're running the two systems, your expert rule-based and statistical-based system, and one of those systems is unable to make a prediction, The fact that there was no structural alerts from that system is not sufficient to then say we met the requirements of the two complementary methodologies and assign it to class 5. So you still have to have a valid prediction from that statistical system. And so what that means is you either run another system, which is our typical practice at FDA CDER, or you can apply expert knowledge to fill that gap and resolve that unknown situation, that data gap. And of course, that application of expert knowledge can also be very helpful for resolving equivocal predictions too. So under the ICHM 7 guideline, applicants are allowed to submit QSAR analyses with any model that's shown to be fit for purpose. And that could be a commercially available model, such as the ones that we use in-house, but it could also be a freely available model, or it could be an in-house constructed model based on a proprietary data set. I think the important thing to communicate here, though, is that if you're using a model that we don't have prior knowledge of, there is an expectation that you will communicate to us how that model was created, all the details of the training set, whether it meets the OECD validation principles, for example. And so there's a higher bar to reporting for models that are not widely used. So when we come to those models that we have no prior knowledge, we expect pretty detailed documentation in order for us to accept the predictions from that model. And one of the formats that's often used to document all the details of a QSAR model is the QSAR model reporting format, or QMRF. So this will show us whether you're actually using an expert rule-based and a statistical-based model together. It will show that the model is trained and predictive of bacterial mutagenicity in all 5 strains of Salmonella, and it will also show that the model is consistent with the OECD validation principles. So our general rule is that we want to trust the predictions that we're getting, but we will verify them in some situations. We do not make a practice of rerunning all predictions that we receive. We only will rerun predictions if there's a concern with what we're seeing in the interpretation or the choice of model, or there truly is just a data gap. So for example, an issue might be that only a single methodology was used, or perhaps no QSAR was used and only visual inspection was used. And that, of course, would be another area where we would need to fill that gap. And then lastly, a situation that does sometimes come up is just where there's an overall conclusion, for example, that the prediction is negative, but the model had obviously identified some structural alert and there's no explanation as to why that alert was dismissed. So those are red flags to us and will lead to some kind of follow-up, and that may lead to us rerunning the prediction in-house. And then when we need to run that prediction in-house, we have our own internal computational toxicology consultation service. So we have a large team of talented scientists who will work up the data, will run the predictions and work up the analyses, apply expert knowledge, and then arrive at a conclusion. And then that's what's used for the regulatory decision making. So in conclusion, I hope I've been able to show you that QSAR models provide a state-of-the-art approach for assessing mutagenicity. They can make a prediction based solely on chemical structure, which is very powerful when we don't have empirical Ames data available for a compound, and they replace visual inspection for the identification of structural alerts. They've been demonstrated to have adequate sensitivity and negative predictivity, which is critical for protecting patient safety, and they've been accepted as being fit for purpose for drug impurities under ICHM 7. Lastly, FDA CDER applies expert knowledge to all internal QSAR assessments because we recognize that it improves the overall performance of the methodology. But we do still continue to see quality issues in regulatory QSAR submissions, and some of the key issues surround the use of appropriate models, in particular the need to have an expert rule-based and a statistical-based model, and we don't always see that. Additionally, we need to know that the models that are being used are consistent with the OECD validation principles, and that may require additional supporting documentation. We need to see that there's been appropriate handling of out-of-domain results, and we also need to see appropriate documentation of assessments, particularly in cases where the model predictions are being overruled based on expert knowledge. And so with that, I would like to acknowledge the many members of our computational toxicology consultation service team who conduct consultations internally, as well as other internal collaborators across the center and our external collaborators that we work with very closely under research collaboration agreements. And thank you for your attention.
Okay, everyone. That concludes the presentation. So at this point, I'd like to open up a Q&A session. Anyone who has a question can go ahead and raise their hand using the hand icon or type their question in the question pane. And Dr. Suman Chakravarti and Dr. Rustam Sayakov will be happy to answer the questions. So let's get started because we did get some questions during the presentation. This isn't exactly related, but someone's asking, what is the acceptable QSAR tool for sensitization endpoint, which can be used for extractables and leachables during a toxicological risk assessment? Rustam, would you be willing to answer that question?
I think, yes, I think the same rules which Dr. Krolak described in her presentations would be applicable. Any QSAR tool which fits the criteria, those OECD principles, and properly validated and ideally well known to regulators would fit. And just as a matter of fact, we do have models which are quite efficient in predicting those endpoints. So you can consider our tool, caseUltra, as one of those tools. But there are some other tools out there too. So the main criteria is even you have your in-house tool, which does a perfect job, as long as you're able to provide all the details describing your tool, you're able to generate QMRF, you probably should be able to share the training set or show the training set. That will be suitable too.
Okay. Where do we keep ICHM7 classification table? The chemical which is mutagenic, but proven to be non-carcinogenic. So I think Naomi answered some of those questions. Correct me if I'm wrong, Suman, about classification and some case studies. But I'd like to let everyone know that we will be actually continuing this discussion in future webinars, discussing more granular information through case studies presented through our scientific team as well as industry contributors. There's actually a handout that's included inside this webinar that you can click on and get more information on the series, which will hopefully provide more detail on the classification process regarding ICHM7 assessment.
Add to it. In Naomi's slides, in one of the latest slides, she had a very nicely formatted table, which you can consider as a summary table when you have a name of the chemical, structure of the chemical with highlighted alerts. Just one word results from statistical rule-based and final conclusion. That would be the perfect place to put those classification results. So you say if it is negative, then you add ICHM7 class 5. If you say it's positive, then ICHM7 class 3. If you know something about carcinogenicity, then appropriate class. I think that would be the best place. If you submitting a report for regulators, that would be the best place.
I mean, it's the important thing that I thought while watching her presentation is to actually have a very nice transparency in the outcome, you know. So it is not a very good idea to just to submit a whole lot of QSAR, raw QSAR reports coming out of the software as it is. If you add, it will be pretty like ambiguous for the person to find out what actually happened. So it is important to add a simple table just to summarize the most important findings at the top, you know, and then that actually gives a bird's eye view of what's inside, and if the regulator finds that there is something that they need to dig into more or investigate a little bit more, they can go into the details. But from a submission point of view, it is a good idea to have a very clear, nice table at the beginning.
Okay. Another question that came in was about nitroso impurities. Could you please provide more insight on nitroso impurities? Yeah, go ahead, Rustam.
Well, there is a phrase, regulatory phrase, cohort of concern means the chemicals of particular importance and probably particular dangers. For those, pretty much everything is known. If you have a nitroso, you most likely going to cause a cancer. So nitroso is one of those which belongs to cohort of concern. That means that whatever M7 says about TTC, it's not quite applicable. You need to go case by case, and you probably need to bridge to known and nitroso compounds with known TTC values. And remember this famous case with Zantac and with this valsartan. So that will give you an idea how those N-nitroso should be handled. You identify your N-nitroso, you either test it because your ICHM7 TTC will not be applicable, or you bridge it to chemicals with known TTC values like dimethyl nitroso or diethyl nitroso, and see if those things are applicable. So those cohort of concerned chemicals that applies not only to nitroso, but there are, I think, 2 more classes. These are something which needs to be taking special attention, and don't take them very lightly. Don't just dismiss them. Most probably it will be pushback from the FDA if you'll do that.
Okay. So another question is concerning the agency not accepting an AIM study conducted on an impurity. So they're wondering why. Why would the agency not accept an AIM study conducted on an impurity?
I can also try to answer that. So the agency likes, and Naomi emphasized a couple of times, the agency likes this AIMS test to be done or evaluated by the book, by the guidance. The crucial guidance for this would be OECD guidance 471, and derivative of that is ICHM7. ICHM7 says you will need to use one statistical, one rule base. Okay, this is about QSAR. But if you're talking about the AIMS results, which are done experimentally, it needs to be done by the guidance. If you submit results only for 2 strains, T98, T100, not good enough. It needs to be 5 strains. Again, it's written in the guidance. If you are not in proper range of concentration, it's supposed to be up to 5,000, I believe, micrograms per plate. It needs to be range of concentration. If you test it only in 1 or 2 concentrations, you at least need to have a reasonable explanation why you did that. If you don't have it, it will be pushed back. Then test needs to be done in triplicate, as far as I remember. If you did not do it in properly, that will be pushed back. So that would be the simple answer. Do it by the guidance, do it in an accredited lab which passed all the inspections, and most likely it's not going to be any pushbacks. But if you did it like in halfway only, that will be noted, and the agency most likely will not accept this experimental data.
And if I can add something there, it has to be done also in the presence and absence of metabolic activation both. Otherwise it's incomplete. So it has to be done.
That's what the guidance demands.
Exactly. Like, we'll go by the guidance, do it with high quality standards. Then there is no reason why they should be not accepting it.
And the guidance again is OECD guidance number 471.
Okay. So someone's asking to please elaborate about the 2-year shelf life comment that Naomi was discussing in slide number 37.
So it's if I can try to answer it, as far as I understand, it's as she says, it's an unofficial rule of thumb. So it's a rough practice they usually maintain, that if you submit a QSAR result done by a software which is not more than 2 years old, then they usually accept it without going into much details into it, But if you submit something that is submit a result, and the QSAR study has been done 2 years ago or more than 2 years ago with a version, or you just did the study more than, let's say, 3 years ago. So what they will do, they will actually investigate more into the results. They will try to redo the QSAR study using the latest version of the software. So usually 2 years is the buffer zone between. During that time, they usually trust the result. So they consider it as recent, But beyond that, they would go into more investigation and check it with the recent versions of the software. That's what I think she meant.
Let me add to that. The reason for this 2-year shelf life is very simple. Every serious software vendor will update the software, will update the models. Sometimes those updates will not lead to drastic change of the results, sometimes they will. Within 2 years period, usually not much changes are happening. But for example, if you will submit aryl baronic acid, which you tested 5 years ago with, I believe, with Derek software, and it will tell you that it's negative, guess what? Your result will not be accepted, because since then there were a lot of changes and aryl baronic acids are now considered to be positive. FDA always has the most updated set of software, so they, from this point of view, they are on the front line. They just, if they don't feel confident in your older results, they will just rerun the results. Most likely, it's not a big deal. There's going to be a paper published soon with a lot of people participating, us included, which says that it was a statistical analysis, how many changes are happening with software during the years of updates. And that more or less coincides with what Naomi said. But just please don't test something and then hold on these 5 years and then try to submit. You most likely will have different results in 5 years.
Okay. So another question that came in, how can we use in-house experimental data in addition to software data to build up our own library? And how do regulators check the validity or acceptance of these libraries?
Well, I think I detailed those descriptions into my workshops back in India, but I can reiterate a little bit that you need to really maintain an in-house database detailing all the experimental details of the aim state that you carry out on your drugs or impurities. So if you are using one of these database outcomes into the analysis of an impurity and submitting to the regulatory people, you just have to submit the relevant study details with the results. You don't have to give them the whole database or something. You just have to add, say, 1 or 2 components, or see analogs, and you have carried out the AIMS test. You have to just add all the relevant details that include what kind of experimental details were used, what kind of strains that has been used on the particular impurity, and related and relevant stuff. So the important thing is to submit the details of the experiment whenever that is relevant.
Let me add to that. It's a great idea to try to build your own models, but quite frequently it's not achievable, because we are using state-of-the-art software, and it's not, well, the best models are obtained when it's not done automatically. That means you need to have a proper expertise, you need to have a proper personnel, you need to have proper tools to build those models. Most likely it's impossible in-house, so you might want to share this data with us. We'll build model for you, but there is an easy solution, at least with caseUltra. Remember that case with Naomi showed the table where she assigned class 4 to one of the compounds based on the known experimental AIMS negative status of one of the compounds later in the table. I don't know which slide it was, but the idea is exactly the same. You have your database of compounds with known AIMS test, and you have your query compounds, like 10 of them, for example, for which you don't know anything. But you know that some of them are similar to those chemicals which you have already tested, and you know that you tested them and they were AIMS negative. So include those 1 or 2 compounds with known experimental Ames negative status into your test batch together with those unknown chemicals and use the consolidator feature. It will help you to mark those known negative, and it will help you to dismiss all potential alerts or out-of-domain status using just this knowledge. In this way, you can use your in-house knowledge without rebuilding the models. I think Tuman will show how to do it in the upcoming series of expert seminars.
Okay, so we'll leave a couple more questions and then we'll wrap up. So I have a question about non-genotoxic API related impurity. If it has a structural alert for a N-nitrosamine, how would it be assessed?
If it is nitrosamine, how it is non-genotoxic? If you know that it is, if it was tested Ames negative, then that's your answer. Your experimental results will overrule any kind of in silico results if it's done properly. If it's tested with proper ranges, with proper strains, with proper protocols, then you use this fact to overrule whatever in silico results you get. If you assume that it's non-genotoxic, but it still contains N-nitroso, you need to do really hard expert review based on whatever analogs you can find to prove that N-nitroso group is not relevant in this case, which I can tell you in advance is going to be very hard because you're dealing with cohort of concern chemical, and to prove that this chemical is negative, that's close to impossible.
I mean, you know, as this is a question that comes, I think, quite frequently about N-nitrosamines, and I have a feeling that whenever N-nitrosamines are involved, actually software plays a very small role, because N-nitrosamines means that it's already known mutagenic concern, mutagenicity concern. You know, it's a very well-known mutagenicity alert and a compound. So the moment you have an N-nitrosamines, then I think the next steps would be to experimentally verify if that's going to be mutagenic or not. Or in that case, suppose you find that, okay, there are a few analogs that shows that in this particular case, N-nitrosamines are not supposed to be mutagenic, that you can include. Another thing that you can include is to show that this N-nitrosamine impurity is not ending up in the final drug. So it's all about experiments, not about actually QSAR anymore, because it's now already known that you got a compound that has an N-nitrosamine functionality, which is a known mutagenic concern.
In our expert system, there is one case which shows that that nitroso group is not necessarily leading to bad results if it's attached to alkane, to see sp3 hybridization carbon, not to nitrogen, to carbon. Nitroso alkanes most likely not going to be potent and mutagens. Anything else you have to analyze.
Yeah, but then it's not N-nitrosamine.
Then it's
just
not
nitrosamine. Yeah.
Yes.
Okay, so one last question. Can you please provide examples of external databases or sites for substructure search?
Well, in our case, we include a very nice database right inside our program that you can actually search using the database, the substructure search tool that we already built in. So you can do that. But from outside, I have no idea, like, if there are any outside database that you can do substructure search on. The main concern is that, if you have such a database, say in the internet or other places, the data is not going to be very reliable. You may not have proper strain results or information or assay information. So it's important to do the substructure search analysis for a database for which you have very nice information available. So that includes our software. I'm sure the other vendors, they also provide some sort of database with their software with tools to search on. But we include such external database right inside our programs.
To add to this, there used to be databases which were dedicated just for genotoxicity, like GenTox. First, it does not have structural information. Second, it was really old database.
Not searching.
Definitely would not support any kind of searching or substructure search. And it's usually like mismatching capabilities and information. If your database will allow you to do substructure search, like PubChem, for example, it will not contain any bacterial mutagenicity data or gene toxicity data. If it does contain gene toxicity data, you probably can search in ECHO database, but you need to know the name. You will not be able to search by structures, by substructures. You can search by structure entire, but not substructure. So it's always a mismatch. The only way to get the product which you really want, which contains all the information which you want, and you can search in any way you want, is to get a commercial software, which probably is well thought and well designed. Well, our software is one of those, and I don't want to advertise any other software of our vendor, of our competitors, which I'm sure you are well aware of, and probably can find similar solutions from them too.
And one more thing to add, suppose you have a database that is proper, and I mean proper means that it contains all the information related to every compound that you have, and you are able to search it by some process. The most important thing to remember the points that Naomi pointed out, that how to search it. So the important thing to remember is that you are trying to solve a puzzle where you are trying to find out if this compound is going to be mutagenic or not. So you need to search around the important functionality that contains the alert, include some features around it, and then search. Then you'll get some hits which might reflect the kind of structural features that you are interested in. So that is the technical aspect of searching, given that you already have a database and you can search in it using substructural queries.
Let me add one more thing, one technicality to this. I just remembered when you were talking about the database. We do in-house use for our internal purposes, at least I use, Chem Office suite, which contains the program which is called Chem Finder. That's actually chemically oriented database manager. So if you get a hold of this software, you'll be able to load your data into it, and you'll be able to do substructural similarity search within it. So you actually can build your chemically meaningful database in-house and use this tool to provide whatever querying you need. But you just need to get the software. So again, it's Chem Office. It's, I think, Cambridge Soft is producing it. I'm not advertising them. I'm just telling you them that we are using them. I'm using them. I'm pretty satisfied with it. Maybe you can find something similar, which is you don't have to pay for it. But this one, it's pretty reliable and pretty powerful.
Okay. So with that, I think I'd like to conclude the session. I invite everyone to, if you have any remaining questions, send them to us via email, and be in touch with either Yatindra or Manoranjan from our Neotel team, and they can relay those questions to us. Otherwise, we will be in touch shortly with more registration links for upcoming webinar series beginning in June.
You can also indicate, Gianna, this handout that you have included in this thing, so that people can see them.
Yeah, so
upcoming ICHM7.
Right. So all of the information for the webinar series is actually included in the handout, which you can see by clicking on the document. But this information will also be sent to you via email, so be on the lookout for that. And we also have recorded this session, so if you'd like a copy of the recording, please let us know and we'll send it over to you. And if anybody else has any concluding remarks, please go ahead. I think we're good. So thanks, everyone.
Right. Thanks, everyone.
Thanks for attending today. Hopefully you found it informative, and we'll see you next time.
Yes, thank you very much for attending, and stay safe, everyone.
Bye-bye.
Bye.